associated with cystinosis. This is a very rare lysosomal storage disease where babies cannot break down cysteine and therefore it accumulates in their cells. If you look at the bottom of the screen, this is the amino acid cysteine. If you link two of these together, you get cysteine. And cysteine often links together to form this structure of cysteine, and you need to be able to break this down in your lysosomes. If you're missing an enzyme, you cannot break this down, and therefore cysteine accumulates all over the body. That's called cystinosis. And one of the first places it tends to accumulate is in proximal tubule cells. So for this reason, babies born with this rare lysosomal storage disease will present in infancy with Fanconi syndrome. Now, just an aside here, don't confuse cystinosis with cystinuria, even though the two names sound alike. Cystinuria is an isolated proximal tubule defect where patients have impaired reabsorption of cysteine. That's that molecule that is two linked amino acids together. The problem here is that cysteine will accumulate in the urine and a rare form of kidney stone called cystine kidney stones will develop. And I talk about this in more detail in the video on kidney stones. Getting back to Fanconi syndrome, let's talk about the acquired causes. So lead poisoning is a classic acquired cause of Fanconi syndrome. Lead can poison proximal tubule cells. Multiple myeloma is also a classic cause of an acquired Fanconi syndrome. In the hematology section, I talk about multiple myeloma, but it often damages the kidneys and it can cause proximal tubule damage, which leads to the Fanconi syndrome. And then finally, there is a long list of drugs that are occasionally associated with Fanconi syndrome. I've listed all of them here. One of the classic drugs is cisplatin, which is used for chemotherapy. Another one is ifosfamide, which is an alkylating agent for chemotherapy. Fanconi syndrome has also been reported associated with these other drugs on the screen here. The next thing I'm going to do is go through this classic graph that you can find in renal physiology textbooks on the screen. It shows the changes in concentration of various solutes as fluid moves along the proximal tubule. So the distance along the proximal tubule is on the x-axis. On the y-axis is the concentration of solutes. However, they are expressed in a very unusual way. They are expressed in the ratio of the concentration of that solute in the tubule compared to plasma. So initially, all of the substances have the same concentration as plasma right after they are filtered at the glomerulus. But then as the tubular fluid moves along the proximal tubule, the concentration changes. Some of those solutes see their concentration rise relative to plasma, others see their concentrations fall. So we're going to try to explain this entire graph in the next few minutes here. The first thing we need to talk about is all the possibilities for how the concentration of solutes might change as fluid moves through the proximal tubule. So let's imagine we have a 50-50 mix of a solute represented by the red dot and water represented by the blue dot. And now let's imagine we move along the proximal tubule in the nephron and we reabsorb the solute, the red dots, at the same rate we reabsorb water, the blue dots. At the end of the proximal tubule, we will still have a 50-50 mix. There's less red and less blue, but the mixture is still 50-50. And this is what happens for sodium and chloride. They are reabsorbed at roughly the same rate as water, so even though we are reabsorbing sodium and chloride, the relative concentration doesn't change much through the proximal tubule. Now let's imagine we have a different solute also at 50-50, and as we move along the proximal tubule, we reabsorb lots of that solute, even more than we reabsorb water. This would leave us with a mix that is less than 50-50. In other words, the concentration of that solute would fall. And this is what happens for glucose and bicarb. These two solutes are actively reabsorbed by the proximal tubule. They are reabsorbed at a greater rate than water, and therefore their concentration falls along the length of the proximal tubule. Finally, let's imagine we have a third mix of 50-50 solutes and water, and we move along the proximal tubule, and let's imagine that none of the solute is reabsorbed at all. In other words, all the red dots remain, but we do lose water as that is reabsorbed in the proximal tubule. Well, now we'd be left with a mixture that has a higher concentration than 50-50, and this is what happens for inulin and creatinine. Inulin and creatinine are not reabsorbed, and therefore they sit in the proximal tubule and become more concentrated as water is pulled out. So now let's go back to this chart and see if we can make sense of it. So glucose, amino acids, and bicarb, they are actively reabsorbed by the proximal tubule, so their concentration falls relative to plasma as shown in this bottom line here. If the reabsorption of a solute were exactly equal to water, it would just be a flat line at 1.0. And you can see that chloride, urea, sodium, and potassium are relatively close to that line. They are reabsorbed at a slightly greater degree than water, but they stay pretty close to the straight 1.0 line if there were no concentration change. Inulin and creatinine follow this straight line up here at an angle because they are not reabsorbed at all. And then finally, PAH, which is para-aminohippuric acid, actually has the steepest line of all. The reason it's like this is because it is secreted by the tubule. So not only is it not reabsorbed, but the tubular cells add PAH to the urine in the lumen, and therefore its concentration rises the greatest. Now we're going to move on from the proximal tubule and talk about the next portion of the nephron, the thin descending loop of Henle. That's this section of the nephron in my drawing here. The thing you need to understand about this portion of the nephron is that it is impermeable to sodium chloride, but it can absorb water, and therefore what it does is concentrate the urine that is found in the tubule. Basically, water leaves the urine, leaving sodium chloride behind, and that makes the urine more concentrated. And the force that draws the water out of the thin descending loop of Henle is the hypertonicity of the surrounding medullary interstitium. So let's talk about that now. Here's a cross-section of a kidney, and this outer portion here is the cortex of the kidney, and that's where you find the glomeruli. However, where you find the loop of Henle is that it dives down from the glomeruli into this portion here, which is the medulla. The outer portion is called the outer medulla, the inner portion is called the inner medulla. And as the urine moves through the tubule down into that medulla, the urine becomes more concentrated, and that's because the solution bathing the nephron or surrounding the nephron in the medulla is very concentrated itself, so it draws water out of the tubule and into the nephron. This is easier to understand with a picture. So here's the glomerulus, and fluid that enters the lumen of the nephron in the glomerulus has an osmolarity of about 300 milliosms, the same as plasma. As fluid moves through the proximal tubule, its osmolarity really doesn't change very much. Even though solutes are being reabsorbed, water is being reabsorbed as well. So fluid that leaves the proximal tubule still has an osmolarity of about 300 milliosms. Then that fluid starts to move down into the thin descending limb, and as it does this, water is being pulled out. And the reason water is being pulled out is because the osmolarity in the outer medulla and the inner medulla is very high. The osmolarity goes up to about 600 in the outer medulla and as high as 1200 in the inner medulla. This is a powerful force for drawing water out by osmosis, and what's going to happen is the fluid in this lumen, when it gets all the way down to the bottom, is also going to have an osmolarity of 1200. The tubular fluid is very concentrated because it's now in equilibrium with the concentrated fluid surrounding the loop of Henle in the inner medulla. It's very high yield for you to understand which solutes are responsible for maintaining that high osmolarity as high as 1200 in the medulla of the kidneys. The three solutes that are responsible for that osmolarity are sodium chloride and also urea. Urea has a very important role to play in the kidneys in terms of maintaining a high osmolarity in the medulla. Urea is generated in the liver by protein metabolism, and I talk about the urea cycle in the biochemistry section. It then travels from the liver to the kidneys where it's reabsorbed by the collecting duct. The collecting duct has a very high permeability to urea, and I'll talk more about how this works later. And then that urea settles into the medulla in the interstitium, and that's essential for maintaining those osmotic gradients. So I'll go through this once again just to make this very clear. Fluid leaving the proximal tubule has an osmolarity of about 300. It's in equilibrium with the cortex of the kidneys. Then as fluid moves down into the loop of Henle, water is removed, drawn out by those high osmolar gradients made by sodium chloride and urea, so that the tubular fluid reaches an osmolarity of 600 when it's in the outer medulla, and then when it gets down to the inner medulla, that fluid is very concentrated as high as 1200. Once urine reaches the bottom of the loop of Henle and has become very concentrated, it then begins to move up the ascending limb of the loop of Henle. And I told you before that the descending limb is permeable to water and impermeable to sodium chloride. The ascending limb is the opposite. It is impermeable to water, however, it is permeable to sodium chloride. What this means is, as the urine moves up this ascending limb, sodium chloride is going to be removed. This is going to pull solutes out of the tubular fluid and dilute it all the way down to a very low osmolarity of 120 milliosms at the top of the ascending limb of the loop of Henle. You'll also notice in this drawing that the ascending limb has two portions, a thin portion and a thick portion. The thick portion is the most important portion. That's where sodium chloride is actively resorbed to take that tubular fluid all the way down to a very dilute and low osmolarity of 120. Sodium and chloride are reabsorbed in the thick ascending limb of the loop of Henle by a very important pump that reabsorbs sodium, potassium, and 2 chloride ions. This is the same pump that is inhibited by loop diuretics, which I talk about in the diuretic module. The reabsorption of sodium is an energy-dependent process. There is a sodium-potassium ATPase pump found in the thick ascending limb. It reabsorbs sodium and creates a low concentration of sodium inside the cell that can be used to drive the activity of this pump right here. Now, some of this potassium that's reabsorbed by this pump will leak back out into the tubular fluid. This creates a positive charge in the urine in the lumen of the nephron at this portion of the loop of Henle. That positive charge is very important because it is used to drive the reabsorption of other positively charged ions like magnesium and calcium. They are reabsorbed via the paracellular route, and they are driven out by the positive charge in the lumen of the nephron. Moving on, now we'll talk about the distal tubule of the nephron. That's this section shown in my drawing down here. It's the section of the nephron that comes right before the collecting duct, which is the last portion of the nephron. This part of the nephron resorbs sodium and chloride. Once again, there is an ATP pump that reabsorbs sodium in exchange for potassium. This creates a low concentration of sodium inside the cell, and that low concentration of sodium is used to reabsorb sodium and chloride. The chloride can then diffuse out a pump on the other side of the cell. The distal tubule is also very important for calcium reabsorption. Calcium can enter distal tubule cells via a channel and then be reabsorbed in exchange for sodium. And this is important because, as I'll talk about in other videos, parathyroid hormone acts on the distal tubule to drive more calcium reabsorption through these channels and transporters. Finally, we get to the collecting duct, the last portion of the nephron. There are two cell types found in the collecting duct. The first cell type is called a principal cell, and the second cell type is called an intercalated cell. The principal cells have an ATP pump that generates a low concentration of sodium inside the cell. In this manner, sodium can be reabsorbed by the principal cells via a channel. This channel is called an ENaC channel. ENaC stands for epithelial sodium channel, and we'll talk about later how this is an important channel because its activity is regulated by aldosterone. In addition, potassium enters the cell via the sodium pump and then can exit into the lumen via a potassium channel. And so in this manner, the collecting duct is important for the secretion of potassium in the setting of high potassium states in the body. And then finally, the principal cells are also important for the reabsorption of water. And I'll talk more about that in a minute because this reabsorption of water is modulated by antidiuretic hormone. The main role of the intercalated cells is to secrete acid. They secrete hydrogen ions into the urine via an ATP pump. So here I've summarized some key points about the collecting duct in terms of its functions. It reabsorbs sodium and water, and it secretes potassium and hydrogen. And then one thing you need to understand is that anytime there's increased sodium delivery to the collecting duct, there will be more potassium excretion. That's because potassium goes out in the collecting duct in exchange for sodium coming in. And more sodium gets reabsorbed when you send more sodium to the collecting duct. So for this reason, hypokalemia often occurs in patients on diuretics like loop diuretics and thiazide diuretics. That's because these drugs send more sodium to the collecting duct, which then gets exchanged for potassium. I'll go back to this slide to make this point clear. So if you send more sodium to the collecting duct, more sodium will enter through this channel and go out through this pump. Then more potassium will come in and go out through this channel, so the two are interrelated. More sodium going in this direction always turns into more potassium going out in that direction, and that's why any drug that promotes diuresis and sends more sodium to the collecting duct can potentially cause hypokalemia. A special feature of the collecting duct is that its activity is heavily influenced by two hormones, aldosterone and antidiuretic hormone. Both of these hormones act on the kidneys and they regulate the function of the collecting duct. Aldosterone is a steroid hormone. It has a structure that is derived from cholesterol, just like all steroid hormones, and this allows it to pass through the lipid bilayer of cells and have effects inside of cells, including on gene expression. It's called a mineralocorticoid because it has effects on minerals like sodium and potassium, and this is in contrast to cortisol, which is a glucocorticoid and has effects on blood sugar. Both cortisol and aldosterone are released from the adrenal cortex, and I talk about this in more detail in the videos on the adrenal gland in the endocrine section. Aldosterone's effects on the collecting duct are summarized on this slide. It can increase the number of sodium-potassium ATPase proteins. It can increase the number of ENaC sodium channels in principal cells. It can also promote potassium secretion by principal cells and hydrogen ion secretion by intercalated cells. Here's a slide showing the effects of aldosterone on the principal cells. The number of sodium potassium ATPases can be increased by aldosterone. Remember, it's a steroid, so it can travel inside the cell and induce gene expression, so it can increase the expression of genes that code for proteins like the sodium potassium ATPase. It can also increase the number and activity of the ENaC sodium channels, and it can also increase the secretion of acid or hydrogen ions from intercalated cells.